Hi everyone, we will continue exploring a new topic, bioinformatics. In this session, we will learn about the digital analogues of nucleic acid processes, transcription, translation, and replication. This presentation is based largely on the information provided on the online book Scientific Computing for Chemists with Python by Charles J. Weiss. In our first session, we explored one of the most common file formats for storing amino acid and nucleotide sequences, the FASTA file. We concluded the session by examining a FASTA file containing DNA sequence of HP1832.2. A FASTA file starts with greater than symbol followed by a descriptive header that provides information about the sequence. The sequence itself follows on subsequent lines. To read a single sequence from a FASTA file, we use the read function. This function extracts key details including identifier, description, name, and sequence data. Identifier, description, name, sequence data. When working with multiple sequences, we should use the parse function instead. This allows us to iterate through multiple sequences using for loop, making it possible to analyze them efficiently. One key analysis we performed was calculating GC content. The fraction of guanine and cytosine base pairs relative to the total base count in the sequence. In session 2, we will dive deeper into digital sequence operation, learning how to manipulate, analyze sequences computationally. Let's take a brief look at the chemical structure of nucleic acid. There are two types: deoxyribonucleic acid, DNA, a double-stranded molecule; ribonucleic acid, RNA, a single-stranded molecule. Both DNA and RNA are made up of nucleotides which serve as their fundamental building blocks. Each DNA nucleotide consists of three components: deoxyribose sugar, 5-carbon sugar without alkyl group at carbon 2, carbon 1, carbon 2. There's no alkyl groups. Phosphate group forms the backbone of DNA strand. Nitrogenous base, one of four types: double-ringed purines, adenine and guanine; single-ringed pyrimidine, cytosine and thymine. Each RNA nucleotide is similar, but a key difference in its sugar. Ribose sugar is a 5-carbon sugar with an alkyl group at carbon 2, 1, 2. Phosphate group forms the backbone of RNA strand. Nitrogenous base, one of four types: double-ringed purines, adenine and guanine; single-ringed pyrimidine, cytosine and uracil instead of thymine in DNA. Each nucleotide is named based on its nitrogenous base. For example, in this diagram, this is adenine, so it is referred as A. DNA is a long polymer made up of nucleotide monomers. Each nucleotide is linked through a covalent bond between phosphate group of one nucleotide and the sugar molecule of the next. This forms a continuous sugar-phosphate backbone as illustrated in the schematic drawing. The nitrogenous bases extend outward from this backbone. DNA consists of two strands, twisted into a right-handed double helix. Within this structure, specific base pairing occurs: adenine and thymine, guanine and cytosine. There are 2 hydrogen bonds between adenine and thymine, and there are 3 hydrogen bonds between cytosine and guanine. Another key feature of DNA is its anti-parallel orientation. One strand has carbon 3 of the sugar facing upward, 1, 2, 3, carbon 3 facing upward, while the other strand has carbon 5, 1, 2, 3, 4, 5 facing upward, ensuring proper alignment for base pairing. In molecular biology, transcription, translation, and replication are essential processes that drive gene expression and DNA duplication. Let's break them down. Transcription, the coding strand of DNA, serves as a template to synthesize messenger RNA, mRNA. This process transfers genetic information from DNA to mRNA, which carries the instructions for protein synthesis. Translation, the messenger RNA sequence is decoded to assemble a protein. In other words, the genetic information transcribed from DNA is now used to synthesize proteins. Replication, each original DNA strand serves as a template to create 2 identical DNA double strands, ensuring accurate duplication of genetic materials. With BioPython, we can easily perform digital simulations of these processes using built-in functions, making it a powerful tool for molecular sequence analysis. BioPython provides several built-in functions to facilitate DNA and RNA sequence processing. Let's go over some key functions. Transcribe function converts a coding DNA strand into messenger RNA, mRNA sequence. Translate function translates a mRNA sequence into a peptide protein sequence. Complement function converts a 5' to 3' DNA strand into its 3' to 5' complementary sequence. Reverse underscore complement underscore RNA function converts a 5' to 3' RNA strand into its 5' to 3' complementary sequence. Complement underscore RNA function converts the 5' to 3' RNA strand into its 3' to 5' complementary sequence. Replace function replaces an existing nucleotide or sequence with a new one. These functions make sequence analysis and manipulation much more efficient in BioPython. Let's walk through the process of converting DNA to RNA and ultimately to a protein sequence using BioPython. The template DNA sequence is first created using the seq function with the sequence in string. The template DNA strand is converted into coding DNA strand, which is the complementary strand in the 5' to 3' direction using the reverse complement function. The transformation follows complementary base pairing rules, changes adenine to thymine, thymine to adenine, cytosine to guanine, guanine to cytosine. Then, the coding DNA is transcribed into messenger RNA using the transcribe function. In this step, thymine is replaced by uracil to match RNA nucleotides. In a biological system, this process is carried out by RNA polymerase, which pairs RNA nucleotides to the DNA template strand. Let's take a quick look at the results, intermediate results. DNA sequence is shown here. It shows that the cytosine has been changed to guanine, cytosine to guanine, thymine to adenine, and cytosine to guanine, and so forth. And the messenger RNA sequence is shown here. The thymine here has been changed to uracil. The messenger RNA sequence is then translated into a peptide protein sequence using the translate function. During this process, each set of 3 nucleotides is converted into a single amino acid, causing the sequence to shrink. Now you can see that the peptide sequence is created. By using Biopython functions, we can efficiently simulate the fundamental transcription and translation processes that occurs in living cells. Now let's run the same code in the Visual Studio Code Editor. First, we create the template DNA using the given sequence. Next, we apply the reverse complement function to convert the template DNA into its complementary strand. Then we use transcribe function to generate the messenger RNA sequence. Finally, using the messenger RNA sequence, we create peptide sequence with the translate function. Okay, let's run the code by clicking the play button. As anticipated, we get DNA sequence, and we get messenger RNA sequence, and we have peptide sequence. Biopython provides powerful tool for performing pairwise sequence alignment on both nucleic acid and proteins. This process helps identify similarities and differences between sequences, which is crucial for biological analysis. The global alignment aligns entire sequence from start to finish, which is best suited for sequences of similar lengths with significant overall similarity. Local alignment aligns subsections of sequences to find common regions between them, which is ideal for identifying conserved motifs or functional domains in longer sequences. How does it work? Biopython uses user-defined scoring rules to evaluate alignments. The algorithm maximizes the alignment score to determine the best possible match between sequences. By leveraging these alignment techniques, researchers can compare genetic sequences, study evolutionary relationships, and analyze protein functions with precision. Now let's dive into the code and see how we can align sequences using Biopython. To perform pairwise sequence alignment, we first need to create a pairwise aligner. This requires specifying the type of alignments, global or local, and optional user-defined score parameters: match score, mismatch penalty, open gap penalty, gap extension penalty. For example, we created an aligner object using the pairwise aligner function. We specified the global alignments with this argument mode equals global. Then we define custom scoring rules. When there is a match, we give 1 point. When there is a mismatch, we give -1 point. When there is a gap opening, there is a -1 point penalty. When there is a gap extension, we give -0.5 penalty. Next, we define two sequences and use the align function to find the optimum alignments. We define the two sequences in string, and we use align function to align these two sequences. Once the alignment is performed, we can check the total number of alignments by printing the length of the alignment list. We get 15 for this particular sequences. We can print each of 15 alignments using a for loop. One of the 15 output is shown in here, and there are 14 more. Finally, we use the score function with the sequences to determine the best alignment score. In this example, the optimal alignment score for the two sequences is 44. This process allows us to compare generic sequences effectively and find the best possible matches. Okay, let's run the same code in the Visual Studio Code editor. This process is essentially the same as before. First, we define the aligner object, which holds both the alignment method and the marking schemes. Next, we introduce two sequences for the pairwise alignment. Then we perform the alignment with these two sequences and generate 15 possible alignments. Finally, we determine the optimum alignment score with these two sequences. Okay, let's run the code by clicking the play button. Okay, the number of alignment is 15 as anticipated, and these are the 15 alignments, and the optimum alignment score is 44 as anticipated. In session 2, we explore the fundamental building blocks of DNA and RNA. For example, DNA consists of three key components: deoxyribose sugar, a 5-carbon sugar lacking a alkyl group at carbon 2, and a phosphate group, and a nitrogenous base, which can be one of 4 types: purines, double-ringed adenine and guanine, pyrimidine, single-ringed, cytosine, and thymine. Each nucleotide is named based on its nitrogenous base. For example, adenine is A, guanine is G, cytosine is C, and thymine is T. The phosphate group of one nucleotide covalently bonds with the sugar of the next nucleotides, forming a long polymer. This creates the sugar phosphate backbone of a single DNA strand, with the nitrogenous bases sticking out from the backbone. DNA consists of 2 complementary strands, 1 and 2, twisted in the right-handed double helix. The nucleotide bases pair specifically. Adenine pairs with thymine with 2 hydrogen bonding, and guanine pairs with cytosine with 3 hydrogen bonds. Additionally, DNA strands are anti-parallel with each other. One strand has its 3' carbon up, while the other has 5' carbon up. We also learned how to perform computational operations on DNA and RNA sequences using BioPython. The transcribe function converts a DNA strand into an RNA strand. The translate function converts an RNA sequence, a RNA sequence into a peptide protein sequence. The align function aligns two sequences based on user-defined optimization rules. These tools allow us to analyze and manipulate genetic sequences digitally, mirroring biological transcription, translation, and sequence alignment. This is the end of this session. Thank you for your attention. I hope you found the session engaging and informative.